Right. This short video will follow up on our videos on lecture 17, where we discuss metabolic integration. So if you haven't watched those videos yet, as well as our video on our BIS103 map number 5, I encourage you to do so first, because in this short video here, what I want to do is just highlight a few examples on how we can apply the aspects of metabolic integration that we discussed in lecture 17 to cancer cells, for example, on how we can use this knowledge of metabolism for diagnosing or treating cancer. And to do so, we actually have to go back all the way to the 19th century and to Louis Pasteur. He actually was a French biochemist. He's most known for the method of pasteurizing milk. That's actually where the name comes from. But he also was very active in the field of glucose and generally carbohydrate metabolism. And one of the observations that he made was that normal cells, so non-cancerous cells or healthy cells, decrease glucose consumption when they are switching from anaerobic to aerobic conditions. So when oxygen comes in. And this makes sense, right? Because if you remember our discussions on ATP yield between fermentation and cellular respiration, right, we had said that under fermentation conditions, we only get 2 ATP out of every molecule of glucose, whereas when we have oxygen, we can run the entire PDH reaction, our TCA cycle and the ETC, and we're getting something around 30 to 32 ATP per glucose. So much, much more ATP per glucose under aerobic conditions. So the idea here being that these cells, when they recognize there's oxygen, they're switching to the aerobic cellular respiration. And so they need much less glucose to achieve the same yield of ATP. Therefore, they're decreasing their glucose consumption. Now, how this related to cancer metabolism now is we have to bring in another scientist, Otto Warburg. He was a German scientist, and he specifically looked into cancer metabolism. And what he observed was that cancer cells, unlike normal cells, healthy cells, they actually maintain lactate fermentation even in the presence of the oxygen. There's some hypothesis, and there's good foundation for that, that this is because cancer cells are so dense that they're really poised to do lactate fermentation because they're often not actually seeing oxygen or are generally oxygen deprived. So their primary metabolism is lactate fermentation. But that also means that they generally have a low ATP yield. Because of that, they're very dependent on high glucose and high glycolytic rates because they're metabolizing very actively and very fast. So they need a lot of glucose, a lot of glycolysis to keep up with the ATP production that is required for that. And because of this observation from Otto Warburg and others, we are actually looking now into some of these glycolytic pathway enzymes and downstream enzymes, and many research groups are working on this, trying to understand if we could use some of these reactions and the dependency on high glucose and the issue of cofactor balance as targets for cancer treatment. Right? One example here is your PDH, right? Your reaction from pyruvate to lactate, right? It requires NAD and NADH. So if you are really dependent on high glucose and high glycolysis and high ATP yields, you have a high demand on your PDH reaction, which makes you very sensitive to losing cofactor balance. And so there's quite a bit of research out there on actually targeting this cofactor balance and the high glucose dependency of these pathways for cancer treatment. So if you're interested in that, I encourage you to just look through some of this research that is published out there for some more details. For our purpose here in the course and our exams, if you understand this general mechanism of how this glucose dependency comes about and how it could potentially be used, that would be perfectly sufficient. It actually already is used in PET scans, right? So we're using the high glucose dependency of cancer cells here for cancer tissue diagnosis using PET scans. And so this really builds on what we call the Warburg effect, so this observation from Otto Warburg. And what people are doing here is that they're using a labeled glucose, right, with a radio-labeled fluoride atom here that is attached to the glucose. So if you're undergoing a PET scan, you will drink the solution with this glucose. And so the strategy here is that because of the higher glucose dependency of the heavily metabolizing cancer cells, these cells will incorporate this radiolabeled glucose much faster and to much higher amounts than healthy cells would. And so if you actually look at this now, here's a PET scan of a patient with, well, this like some lung cancer, and he had probably some cancer in the lymph nodes before chemotherapy, and now here you see it after. So you can use PET scans for this purpose of actually visualizing cancer cells. Because they have this higher glucose dependency, they're incorporating this radiolabeled glucose much more efficiently. You also see it here in the kidneys and in the bladder. These are not cancerous, but these are the major secretion pathways for anything that we're taking in as dietary food, including this radiolabeled glucose. So they do show up there because they're accumulating there. But what you're really focusing on here are other tissues. So it can be used, right? This knowledge of glucose and carbohydrate metabolism can actually actively be used to develop diagnostic tools.